Atoms are the fundamental building blocks of matter, consisting of protons, neutrons, and electrons. Protons, which are positively charged, and neutrons, which have no charge, form the dense nucleus at the center of an atom. Electrons, carrying a negative charge, orbit the nucleus in energy levels or electron clouds. The number of protons in the nucleus, known as the atomic number, defines the element and its chemical properties. Elements are pure substances made up of only one type of atom, and they cannot be broken down into simpler substances through chemical means. Each element, such as hydrogen with 1 proton or oxygen with 8, has unique characteristics. These elements, organized in the periodic table, combine to form compounds and molecules, which make up all the matter around us. Elements can be classified as metals, nonmetals, or metalloids based on their physical and chemical properties. Together, atoms and elements form the basis for understanding chemical reactions and the composition of the physical world.
Molecules are created when 2 or more atoms bond together chemically, forming the smallest unit of a compound that retains its chemical properties. These bonds between atoms come in several forms, with covalent bonds being the most common, where atoms share electrons to achieve stability, as seen in water, H2O. Another type is the ionic bond, which occurs when atoms transfer electrons, creating charged ions that attract each other, such as in sodium chloride, NaCl. A third, weaker bond, called a hydrogen bond, occurs when a hydrogen atom, already covalently bonded to one electronegative atom, is attracted to another electronegative atom. These bonds, though weaker, play a vital role in the structure of large molecules like DNA. Whether through covalent, ionic, or hydrogen bonding, the interactions between atoms allow the formation of molecules, which serve as the foundation for everything from simple substances to complex biological systems.
Water is essential to all life on Earth, playing a vital role in biological processes and the survival of organisms. Its unique chemical properties, due to the structure of water molecules, H2O, make it indispensable. Water molecules are polar, with a slight positive charge on the hydrogen atoms and a slight negative charge on the oxygen atom, allowing them to form hydrogen bonds with each other. These bonds give water many of its key characteristics, such as its ability to dissolve a wide range of substances, its high heat capacity, and its cohesive and adhesive properties. Water's ability to dissolve other substances makes it an excellent solvent, which is crucial for transporting nutrients, waste, and gases in biological systems. Inside cells, water acts as a medium where biochemical reactions take place, allowing enzymes and other molecules to function efficiently. Water also plays a role in regulating body temperature due to its high specific heat, meaning it can absorb and release heat with minimal temperature change. This property helps maintain stable environments for organisms, whether within their bodies or in ecosystems. Moreover, water molecules sticking to each other, and adhesion, water molecules sticking to other surfaces, enable vital processes like capillary action, which allows plants to draw water from their roots to their leaves. Its role in photosynthesis is another key contribution, as water provides the electrons needed for the energy-converting reactions in plants. Without water, life as we know it could not exist. It is fundamental for cellular function, temperature regulation, chemical reactions, and nutrient transport, making it the foundation of all biological systems on Earth.
Acids and bases are 2 types of substances that play crucial roles in chemistry, particularly in biological and chemical processes. They are defined by how they interact with hydrogen ions, H+, in a solution. An acid is a substance that donates hydrogen ions, H+, when dissolved in water, increasing the concentration of H+ in the solution. This gives acids their characteristic properties, such as a sour taste, the ability to turn blue litmus paper red, and their reactivity with metals. Examples of common acids include hydrochloric acid, HCl, found in stomach acid, and citric acid found in citrus fruits. Acids range in strength, with strong acids like sulfuric acid, H2SO4, completely dissociating in water, while weak acids like acetic acid, Ch3Cu, partially dissociate. A base, on the other hand, is a substance that accepts hydrogen ions, H+, or donates hydroxide ions, O-, when dissolved in water. This results in a decrease in H+ concentration, making the solution more alkaline. Bases are often characterized by a bitter taste, slippery feel, and the ability to turn red litmus paper blue. Common bases include sodium hydroxide, NaO, used in soaps and cleaning products, and bicarbonate, HO3-, which is found in baking soda. Bases, like acids, can also vary in strength, with strong bases like NaO fully dissociating in water and weak bases like ammonia, Na3, partially dissociating. The pH scale measures the acidity or alkalinity of a solution, ranging from 0, most acidic, to 14, most basic, with 7 being neutral, pure water. Acids have a pH less than 7, while bases have a pH greater than 7. The balance between acids and bases is critical in many biological systems, such as maintaining the pH of human blood, which is tightly regulated around 7.4. In summary, acids donate hydrogen ions and have a pH below 7, while bases accept hydrogen ions or donate hydroxide ions and have a pH above 7. Both are essential for various chemical reactions and biological processes, from digestion to maintaining homeostasis in living organisms.
Organic molecules are the foundation of all living organisms, consisting primarily of carbon atoms bonded to hydrogen, oxygen, nitrogen, and occasionally other elements. Carbon's ability to form 4 covalent bonds makes it uniquely suited to create complex, diverse structures necessary for life. The 4 major classes of organic molecules, carbohydrates, proteins, lipids, and nucleic acids, each serve specific, crucial roles in biological systems. Carbohydrates, composed of carbon, hydrogen, and oxygen, are the primary energy source for organisms, with glucose being a key fuel, while complex carbohydrates like starch and cellulose provide energy storage and structural support. Proteins, made of amino acids linked by peptide bonds, perform countless functions including catalyzing reactions as enzymes, providing structure to cells and tissues, and regulating processes as hormones. Proteins are central to nearly all biological activities, thanks to their complex, folded structures. Lipids, including fats, oils, and phospholipids, are hydrophobic molecules responsible for long-term energy storage, forming cell membranes, and acting as signaling molecules like steroids. Their structure, with a high carbon-to-hydrogen ratio, makes them energy-dense and crucial for maintaining cellular integrity. Lastly, nucleic acids, like DNA and RNA, are the molecules that store and transmit genetic information, with DNA encoding the instructions for building and maintaining an organism, and RNA translating that genetic code into proteins. Together, these organic molecules interact to form the chemical basis of life, supporting the complex processes required for growth, reproduction, and adaptation in living organisms.
Cells are the basic units of life, forming the structural and functional foundation of all living organisms. They are highly organized, self-contained units capable of carrying out essential life processes such as energy production, waste elimination, reproduction, and cellular communication. To observe cells and their intricate structures, scientists use microscopes. Light microscopes allow us to see the general shape and size of cells, while electron microscopes provide much greater magnification and resolution, enabling us to examine subcellular structures like organelles in stunning detail. One key concept in cell biology is the surface area to volume ratio, which explains why cells remain small. As a cell grows, its volume increases faster than its surface area, which limits the cell's ability to efficiently transport nutrients and remove waste. This size limitation is why most living organisms consist of many small cells rather than fewer large ones.
Despite their size differences, all cells share some fundamental characteristics. All cells have a plasma membrane, a semi-permeable phospholipid bilayer that regulates the entry and exit of substances, ensuring homeostasis. Inside the cell, the cytoplasm provides a gel-like environment where cellular reactions take place. Additionally, all cells contain DNA, which holds the genetic blueprint for the cell's functions, and ribosomes, which synthesize proteins based on genetic instructions. However, cells can be divided into 2 broad categories: prokaryotic and eukaryotic. Prokaryotic cells, found in bacteria and archaea, are relatively simple in structure, lacking membrane-bound organelles and a true nucleus. Their genetic material resides in a nucleoid region, and they are often much smaller than eukaryotic cells. On the other hand, eukaryotic cells, which make up plants, animals, fungi, and protists, are more complex and contain numerous membrane-bound organelles, each with specific functions.
In eukaryotic cells, the nucleus acts as the control center, housing DNA and overseeing the cell's activities by regulating gene expression. The mitochondria, often called the powerhouses of the cell, generate ATP, adenosine triphosphate, the cell's energy currency, through cellular respiration. The endoplasmic reticulum, ER, which comes in two forms, rough, with ribosomes, and smooth, without ribosomes, is critical for synthesizing proteins and lipids. The Golgi apparatus modifies, sorts, and packages proteins and lipids for transport to different parts of the cell or outside the cell. Lysosomes act as the digestive system of the cell, breaking down waste materials, while peroxisomes detoxify harmful substances. In plant cells, chloroplasts are vital for photosynthesis, converting sunlight into chemical energy stored in glucose. The structural integrity of plant cells is further supported by a rigid cell wall made of cellulose, which provides strength and protection.
All of these eukaryotic organelles work together in a highly coordinated manner to maintain cellular function and enable complex life processes. The cell's internal framework, known as the cytoskeleton, gives it structure, facilitates movement, and plays a key role in intracellular transport. Composed of microtubules, microfilaments, and intermediate filaments, the cytoskeleton helps cells maintain their shape and assists in the movement of organelles and vesicles within the cell.
The cell membrane is another critical structure found in all cells. It not only acts as a barrier separating the internal environment of the cell from the external surroundings, but also plays a key role in communication and signaling. The membrane is selectively permeable, allowing some substances, such as water and oxygen, to pass freely, while blocking others. Embedded proteins in the membrane facilitate the transport of larger molecules or ions that cannot easily pass through the lipid bilayer. The cell membrane also helps maintain homeostasis, a state of balance within the cell, by controlling the movement of ions, nutrients, and waste products.
In summary, cells are incredibly complex, self-sustaining units that carry out the essential functions of life. Whether prokaryotic or eukaryotic, they share basic features like a plasma membrane, cytoplasm, DNA, and ribosomes. However, eukaryotic cells distinguish themselves by containing specialized organelles such as the nucleus, mitochondria, ER, and Golgi apparatus, allowing them to perform complex functions and support multicellular life. The cell membrane plays a crucial role in maintaining homeostasis and enabling communication with the external environment. Together, these components make cells the building blocks of all life, from the simplest bacteria to the most complex multicellular organisms.
Complementary base pairing is a fundamental principle of molecular biology, referring to how nitrogenous bases in DNA and RNA pair with each other. In DNA, adenine, A, pairs with thymine, T, and cytosine, C, pairs with guanine, G, through hydrogen bonds, forming the rungs of the DNA double helix. In RNA, thymine is replaced by uracil, U, so adenine pairs with uracil instead. This pairing is crucial for the processes of transcription and translation, ensuring accurate replication and expression of genetic information.
Transcription is the process by which the genetic information in a DNA sequence is copied into a complementary RNA sequence. This process begins in the nucleus of eukaryotic cells when the enzyme RNA polymerase binds to the DNA template strand. RNA polymerase moves along the DNA, reading the sequence of nucleotides on the template strand and synthesizing a strand of messenger RNA, mRNA, based on complementary base pairing. During transcription, the DNA's template strand is the strand that is read by RNA polymerase to build the RNA molecule, while the non-template strand is not directly involved in transcription but has a sequence identical to the RNA, except for uracil replacing thymine.
RNA polymerase is the key enzyme responsible for synthesizing RNA during transcription. It binds to specific regions on the DNA called promoters and unwinds the DNA helix to expose the template strand. RNA polymerase then catalyzes the formation of RNA by adding complementary RNA nucleotides, A, U, C, and G, to the growing mRNA chain. The RNA polymerase continues elongating the RNA strand until it reaches a termination signal, at which point transcription stops and the mRNA strand is released.
In transcription, the template strand of DNA is the strand that is actively used by RNA polymerase to synthesize the RNA molecule. It serves as the blueprint, with RNA polymerase reading the nucleotide sequence and adding the complementary RNA bases. For example, if the template strand has a sequence of ACGT, The corresponding RNA sequence will be euka. The non-template strand is sometimes called the coding strand because it has the same sequence as the RNA transcript, except thymine is replaced by uracil. It does not participate directly in the transcription process. In transcription, the template strand of DNA is the strand that is actively used by RNA polymerase to synthesize the RNA molecule. It serves as the blueprint, with RNA polymerase reading the nucleotide sequence and adding the complementary RNA bases. For example, if the template strand has a sequence of ACGT, the corresponding RNA sequence will be euka. The non-template strand is sometimes called the coding strand because it has the same sequence as the RNA transcript, except thymine is replaced by uracil. It does not participate directly in the transcription process. Ribosomes are complex molecular machines composed of both proteins and ribosomal RNA, rRNA. They consist of two subunits, a large subunit and a small subunit. Ribosomes play a central role in translation by binding to the mRNA and facilitating the assembly of amino acids into a polypeptide chain. During translation, transfer RNA, rRNA, molecules bring specific amino acids to the ribosome, where they are added to the growing protein chain based on the codon sequence of the mRNA.
rRNA, ribosomal RNA, is a type of RNA that forms a key structural and functional component of ribosomes. rRNA not only helps maintain the shape and stability of the ribosome but also plays a crucial enzymatic role in catalyzing the formation of peptide bonds between amino acids during protein synthesis. Although ribosomes contain several proteins, rRNA is essential for their function, as it acts as the site of translation and ensures the accuracy of the process by helping to align rRNA and mRNA properly.
In summary, complementary base pairing ensures accurate transcription and translation of genetic information. RNA polymerase transcribes DNA into mRNA by reading the template strand, while the non-template strand remains inactive. The mRNA is then translated by ribosomes, which consist of proteins and RNA, and this process results in the synthesis of proteins that are crucial for cellular function.